Welcome to AP Biology. Today we're going to be looking at hypothesis testing and chi-square analysis. This is a statistical test that you will be asked to do sometimes for AP Bio, and you'll also regularly have to interpret what chi-square value results mean. So we'll do some practice with this, but let me just get into the definitions and be patient with yourself as you go through this because I think it does take some practice to really get it. So hypothesis testing is procedures used by scientists to either reject or fail to reject a statistical hypothesis. We'll define what I mean by a statistical hypothesis on the next page, but when you do experiments, you're going to get some variation. Is that variation within a range where you would kind of still think that what you're expecting is correct, or maybe your variation is so big that what you were originally thinking is actually not true? So we have two hypotheses, and you should memorize what a null hypothesis is and what an alternative hypothesis is. Sometimes we abbreviate it with an H0 for null hypothesis. Null means nothing. There's nothing going on here. So the null hypothesis is the hypothesis that there's no difference between what you observe and what you expect to find. Your alternative hypothesis is the exact opposite. It's that your observations are the result of some non-random cause. There is not nothing going on. Your observations are different than what you had expected, or at least your theoretical outline of what you had expected. Your hypotheses should be testable, measurable, clear, all of that. Okay, so here's an example. Let's say you have a die and you're rolling it, maybe you roll it 1,000 times, and you're thinking that this, or at least that what you're presenting is that this die has it's not weighted on one side or the other. So you roll it a whole bunch of times. Your null hypothesis is that there's nothing funky going on here. You're going to expect 1/6 of the time to get ones, 1/6 of the time to get twos, etc. They should all be equal. Your alternative hypothesis is that you're not going to get all of them to be 1/6. At least one of them isn't that way. Maybe it's actually a heavy die. It's weighted so that you're going to get sixes more often. Here's another example. When you have a sample, usually there are different variations in your sample than the actual variations in a population. The bigger your sample is, the more likely it is that your variations will reflect the population. But at what point do you say this sample actually isn't part of the population? It's so different from what I expected that it's actually probably part of some other population instead. Or maybe there's been enough natural selection that the population is now different than what it was when I tested it 30 years ago. All right, so here's what the chi-square analysis looks like. Let's define it first. It's a statistical method used to compare two groups of data. We're going to call those two groups of data the observed data, which is what you do in your experiment or what you count out in the field, versus expected, which is your theoretical expectation. Most of our chi-square analyses are going to be done on genetics crosses, so I'll do some practice with you on that. So let's say you have some fruit flies and you're thinking that the red eyes versus, I don't know, some other maybe brown eyes, maybe you're expecting red eyes to be dominant and it's not on a sex chromosome, so it's just, we're expecting just normal autosomal dominance, if you remember what that is. If you don't, we're going to do a whole unit on genetics, so don't worry about it too much. But if I do this cross and get several thousand babies, then I know I'm not going to have like exact numbers, but how close, like let's say I get my observed fruit flies, the babies, as a result of some cross, how close is close enough to say that, yeah, it really is red is dominant to brown, right? So is the variation between observed values and expected or theoretical values, is it due to chance? Because sometimes you flip a coin and you don't necessarily always get heads 50% of the time, right? So is it just due to chance, or is there some real difference going on here? All right, so one other thing that my students mess up all the time, which is so easy to mess up, this thing is the symbol for chi-squared. You don't take the square root of this. It's just once you do your calculation, you've got the numbers. So let's do a practice calculation, and then we'll have to go through the notes so I can show you what the calculation even means. So let's say we have a cross between red eyes and brown eyes. So I'm going to say big R is red. With my model, little r I'm going to say is brown. and I'm going to say it's not on an X or a Y chromosome. So let's say I have a heterozygous, a fruit fly with red eyes, but I know its mom had brown eyes or whatever, so I think it's heterozygous. And I'm mating that with a brown-eyed one. And let's say I do that. If I do that, here's my cross. This one has a gamete that's big R, that has a gamete that's little r. And then I can maybe do this. And so the genetics crosses get more complex than that, but let's just do this one for now. So I'm expecting that 50% of my babies will have red eyes, right? These are going to be red. And I'm expecting that 50% of my babies will have brown eyes. Well, of the fruit fly babies, right? So I'm going to have the observed babies. And so let's say I do this and I end up with 2,943 babies. Well, 50%, let's see, let's do, let me get some numbers here. Okay, let's say, I don't know, let's say 1,690 of them have red eyes. And let's see, 2,943, whoops, 2,943 minus 1,690. That means that the ones that had brown eyes, and this is observed means that's what I actually counted up. So 1,253 of them had brown eyes. Okay, so when I do the cross between this one, and I do that cross a bunch of times, and I count up all the babies, this is what I get. So the observed is actually what I count, right? That's actually that's actually what I count up. My expected is the theoretical. So if I have 2,943 of those times 0.5, I'm going to get, I just did that wrong, 2,943 divided by, whoops. Okay, so, okay, divided by 2. So 1,400, and let's say 72 of them I'm expecting to have red eyes. And I'm expecting that same number of brown-eyed. They're not going to actually add up to this exactly because that's odd, but it's going to be close enough. You can actually do the 1471.5 if you want, but since we don't get fruit flies in half numbers, I'm just rounding it up. Okay, so the first thing that I do, remember, so this thing right here means sum. That's what I do at the end. First, I'm going to do this. So if you remember your order of operations, parentheses is going to come first. So observed minus expected. So I'm going to get 1690 minus 1472. And that gives me 218. And 1253 minus 1472. Whoops. 1253 minus 1472 is going to give me 219. Negative, and I don't really care if it's negative because the next thing that I'm going to do is square it. So I'm going to take that number and I'm going to square it. So that gives me a really big number, 47961. Okay, so this number 47524. All right, and so then up here, the next thing you need to do is divide by E. So expected is 1472. So I'm going to take this number and divide it by what I expected to get. 1472, and I'm going to get 32. Let's do 32.29. And then this one is 47961. And then I want to divide that by my expected, which is 1472. and I'm going to get 32.58. And then the next thing to do is add them up. So I'm going to add those two numbers. 32.29. I get 64.87. Okay, so that's my chi-square value, and I'm just going to write it like that. I'm going to say my chi-square value is this, and I don't take the square root. I just leave it as is. What does that number even mean? Well, I don't know. So what it means is the important part. So let's talk through the rest of the chi-square, and then we'll come back to this to say whether these numbers are close enough to these numbers to call them, to say that what I saw actually follows what I think I should have seen. All right, so this chi-square analysis is used to see if a sample is really the same as a particular population. That's called goodness of fit test. You're checking to see if your results match up to some known or theoretical outcome. So is what I observed with the fruit flies what I actually expected to get with the fruit flies? You need a p-value, which is going to be for us 0.05, could be 0.01 sometimes, but usually we're going to use a p-value of 0.05. This is a calculated probability. It's a probability of finding the observed results when the null hypothesis of a study question is true. So when we, like the other, back in this problem where I said there were 1496, what was it, 1472. So 1472 should have been brown-eyed or whatever. We're going to pick a critical value so that you could get numbers outside of your range, but they're going to end up only 5% of the time outside of the range. So you have to pick some number to cut off with. We're going to pick a value of 0.05. That means that it sort of means that 95% of the time that we do this test, we're going to get numbers that are very close, that are accepted. It doesn't really mean that, because the opposite of a null hypothesis, like the alternative hypothesis, there could be other alternative hypotheses, but that's what we're going to go with. Okay, so the p-value, or calculated probability, is a probability of finding the observed results when the null hypothesis is true. I said that already. We're going to pick 0.05. Occasionally, they'll ask you to use a 0.01. I'll show you what that means. The critical value is the number that you compare your chi-square result to determine whether to accept or to reject the null hypothesis. So if the chi-square value is greater than the critical value, then there's a lot of variation. So if that chi-square value that you calculated, remember we calculated a chi-square value of like 64 or something back a few slides. We're going to compare that to some critical value, and that's going to be your cutoff. That's going to say whether you have too much variation or if it's a little bit of variation. So if you don't have too much variation, then this number is going to be small. But what's a small number? Is 64 a small number for a chi-square value? I don't know. We'll compare it to some critical value, and we'll have a table to look it up with. So if your chi-square value is greater than the critical value, then that means you calculated a lot of variation. There's a lot of variation. And so you're going to reject your null hypothesis, and you're going to say, no, the variation is not due to chance. There's something else going on. If the chi-square value is smaller than your critical value, then there's not much variation and you accept your null hypothesis. So there's no difference between the populations, and your variation is just due to chance. So I'll show you an example of that too. Okay, so on your AP formula sheet, there's something that looks like this, and it will be near your chi-square formula. So this says right on it, chi-square table. Here are the degrees of freedom. So what does all that stuff mean? So it says right on your formula sheet, degrees of freedom are equal to the number of distinct possible outcomes minus 1. So in our example, we had red eyes and we had brown eyes, and you got a certain amount of variation. If you also had sepia eyes and black eyes and purple eyes and white eyes or something, you're going to expect just to get a bigger number because of that. So we have different critical values. These things are critical values. We're going to have different critical values based on how many different categories you have. So the categories is really the degrees of freedom sort of minus 1. So your degrees of freedom are equal to the number of distinct possible outcomes minus 1. So we had 2 outcomes, red eyes and brown eyes. So that's 2. So 2 minus 1 is 1. So we, for this example, are going to use a degree of freedom of 1. I'm going to tell you to use a p-value of 0.05, meaning there's only a 5% chance. Meaning that there's only a 5% chance that your variation isn't caught within this number. So your critical value at a p of 0.05 is the amount of variation you would expect 95% of the time. So take a look right here, 3.84. That's my critical value. So do you remember how I calculated a chi-square value of, I think it was 64. Let's go back and see. Yep, our chi-square value was 64, right here, right? So I'm comparing my chi-square value of 64 to this table, and I'm comparing it, look at my numbers, 3.84. So my chi-square value is much, much bigger. So that means I had way more variation than I would expect if the number of red-eyed fruit flies was equal to the number of brown-eyed fruit flies. And you can kind of see that when you look at your numbers. 1690 when it should be 1472, or 1253 when it should be 1472. That's a lot more variation than if I just flipped a coin 2,943 times, right? So there's something going on here. These numbers aren't close enough to 50-50. So when that happens, I'm going to say I reject my null hypothesis that there's no difference between observed minus expected. These red versus brown are not even. I didn't get 50-50. You can also use a chi-square value of p equals 0.01. This is the amount of variation you would expect 99% of time. So 99% of the time, if you have a chi-square value smaller than 3.84 in this case, then you're going to get your variation in. Yes, sometimes you might get more variation, like 1% of the time, but we've got to make the cutoff somewhere. Okay. So let's say instead you did a different genetics test and you had 4 outcomes. You had a red-eyed, black-bodied fruit fly, a red-eyed, white-bodied fruit fly, a brown-eyed. What were my different choices? So let's say you have red eyes and ebony body. Or you could have red eyes and let's go with wild type body, whatever. I guess a lighter colored. Or you can have white eyes. No, actually I was using brown. Let's use brown. Brown eyes with ebony body. Or they could have brown eyes with wild type body. Those are 4 different possibilities. And so now I have a degree of freedom of 4 minus 1, which is going to be 3. Notice that my critical value now is 7.82. I'm going to expect more variation just because I have more categories. So when you're doing these, start with a degree of freedom, pick your p-value. We usually use 0.05 unless they tell you to use 0.01. This takes in 95% of the variation. Find your critical value, memorize that. These things in here, these numbers are critical values. You compare your calculation to the critical value. Let's say I do a cross and I'm expecting a 1-to-1 to 1-to-1 ratio here. And let's say I get a chi-square value of 5.2 here of my observed flies versus what I expect with the 1-to-1 to 1-to-1. I'm going to take that 5.2 and I'm going to go over here. I've got 4 categories. 4 minus 1 is 3. I've got 3 degrees of freedom, so my critical value is 7.82. I would say that my chi-square value is smaller than my critical value. Therefore, I accept my null hypothesis that there's no difference between what I observed and what I expected. Any variation that I got here between these that wasn't exactly 1-to-1 to 1-to-1 is just random variation. Okay, so I sort of said this already. Degrees of freedom, this takes into account the fact that the value of the chi-square is calculated as the sum of the squared deviations for all the classes. So you have a natural increase in the value anytime you have more categories added in. Okay, we're going to do a lab with either M&Ms or Skittles together in class. So we'll do that together as a practice. AP really wants you to write this stuff out clearly, exactly the way they want it, or more or less the way they want it. So let's say you have a chi-square value of 0.05. So you did all your math and you ended up with your chi-square value equal to 0.04. And you had 2 categories, so that means your degrees of freedom is 1. And so your critical value is 3.84. So here's what they want you to say, or here's the way I want you to write it. The critical value from the table with 1 degree of freedom and a p-value of 0.05 is 3.84. Did you see that? The critical value, that's this, from the table with 1 degree of freedom, p-value of 0.05, they come together at 3.84. The variation that I calculated, chi-square of 0.04, using a chi-square is much lower than the critical value of 3.84. Really low, meaning you don't have much variation. So I accept my null hypothesis, there's no difference, and they are the same population. Any variation is just due to chance. Practice writing this out. They want it written like that. Let's say you do another example and you come up with a chi-square value of 10.78. So here we go. So your chi-square value is 10.78. You have 5 categories, so that means your degrees of freedom would be 4, right? 5 minus 1. And so if you look at that, 4 degrees of freedom, I didn't put this chart in there. So with 4 degrees of freedom, I have a critical value of 9.49. And that's what it says right here. So the critical value from the table with 4 degrees of freedom and a p-value of 0.05 is 9.49. That's what I would actually write on my answer. The variation that I calculated, 10.78, that's the chi-square value, using chi-square is higher than the critical value. That means you have more variation, right? So I reject my null hypothesis. There are different populations. The variation is not just due to chance. I accept my alternative hypothesis, which means that there's something different, right? So if my null hypothesis was that I'm getting a 1 to 1 to 1 to 1 ratio, but I get this chi-square value, I have to reject that. It is not a 1 to 1 to 1 to 1 ratio. My alternative hypothesis is that it's not this. There's something else going on. So now I'll do some more experiments to try to figure out what else is going on.